First of all, as introduced by my dear colleagues, myself, Dr. Ganpati Mukri, a senior scientist working in Division of Genetics on particularly maize breeding. So in this particular topic, I would like to discuss majorly on the cob length. That is one of the important trait in maize breeding, cob length as well as kernel row number. Some of you have already might have the experience of maize or maize breeding, or maybe some of you are new to this. So for them, I can say like this: the length of the cob decides the number of kernels per plant, and as well as the number of the kernel row number decides the weight as well as the size of the kernel uh, in the given crop in maize particularly. So in that there are so many genes are responsible for this the trait expression. All of you might have know this. For every trait expression there is a gene which is responsible for expression of the trait finally, which decides maybe the yield in terms of phenotypic expression. In that way, so I would like to discuss what are our effort in understanding this kn1 gene, that is particularly known as knotted 1 gene, which is responsible for deciding the cob length in tropical maize particularly. One of my topic says tropical field corn because these studies already been done in temperate background, but minimum understanding is there in case of tropical field corn. So we try to understand this particular the role of this particular gene in our maize and how we have utilized this particular gene in our maize breeding. So it may be like in any kind of agriculture, say it may be natural farming or whatever. So, but yield is our major target, and since the yield is a very complex trait, we have to understand the component traits first. So with this background, I will proceed further. So maize, all of you know that is one of the important cereal crop after wheat and rice. It is extensively grown in India, covering around 11 million hectares approximately, with the productivity of 3.7 tons per hectare. So though uh, recently the maize yield has been improved over a period of time, but still there is a gap of around 2 to 3 tons when you compare to the world productivity or international productivity, which is around 6 tons, 5.75 tons per hectare. So in that way, there is a scope to improve this maize yield, particularly yield per se of the particular is our concern. So to match with the international production productivity. And you know that maize is consumed in a wide range of array in case of in India as well as in abroad also. So that may be used for starch extraction purpose as a cattle feed in majorly, and also in human direct consumption in terms of maybe normal culinary purposes, and also poultry feed major of the time. Nowadays, it has been having tremendous utilization in ethanol extraction also. But for all these things, yield is one of the important criteria for which farmers are eagerly waiting for higher productivity. When yield enhancement is there, then it will have on a marketing place also. So that is one of the important criteria where maize has been targeted for breeding. So as you know that I told you, like there are different component traits of yield. So we cannot improve as yield as such in one go. So we have to target one trait at a time to improve the particular uh, trait of interest for any trait for that matter. But yield is like it is a quantitative trait which is composed of many component traits which will influences the ultimate final yield of the maize. So for example, like a cob girth, one of the important trait which supports the number of kernels on the each cob. Cob, you know that it is the major part, edible part or grain supporting part. It is cob. The cob length is one of the another important component traits which will decides how many kernels can fit into the cob. And kernel row number, it will decides number of kernel rows like this. This is the number of kernel rows, which also indirectly decides number of kernels, total number of kernels which can accommodate by the cob. And maybe kernels per row, kernel weight, and all those things will be matter. They are indirectly one depend on each other. When kernel row number is increases, there may be slightly reduction in the kernel weight or kernel size. But when you increase the kernel length, cob length, so that it will give again the space for accumulation of this kernel row, so kernels, so that the yield can be stabilized or maintained. So that means this cob girth, cob length, and kernel numbers. Kernel numbers means 1, 2, 3, 4. That is, this is the kernel number. These are all are interrelated, and then ultimately that will decides the yield. That we can see that there is a correlation. We can analyze this statistically also. So there is a strong correlation between these traits. That means you can target any one of these traits to tackle the deficiency of other traits. If you increase the cob length, that means cob seed size will be taken care. If you increase the kernel row number, that more number of kernels can be accommodated in the per crop. Finally, good yield can be expected. So this is our target actually. For that purpose, we studied two traits, particularly kernel row number as well as cob length of the cob in our study. So as you know that as I told you, like there are different rows patterns. Huh? So that you can see the particular genotype AI517 having 18 kernels, AI505 having 12 kernels, AI521 having 10 kernels. There are different kind of genotypes are there. These are decided by some of the other genes. Like there is a pathway which decides, the SAM pathway, pseudoapical meristem pathway we can say. It is scientifically a Ussel clavata pathway that will decides the how that kernel row number is decided. SAM pathway will further finally decided by the spikelet meristem pathway. The spikelet meristem will be decided in the variation in the kernel source. You can see from the starting from the 10 to 18 kernel row. This will be decided at the genetic level, that is through the SAM pathway. So we studied in our genetics division or IRA. So there are ranges of kernel row number genotypes are available. They're starting from 18 to more than 20, more than 20 or 22, and so. So there is a genetic reason for this. We try to understand this in our study. So what we did was, like, we had taken different kinds of genes available which can control the kernel row number, that is FE2, FE4. These are all the genes. So I think range of audience, maybe even from students also. For students is that you can know that there are different genes which are responsible for some traits, or single gene can contribute to the many deciding many traits also, or many genes can contribute to the single trait also. There are so many possibilities. Here in this case, there are different genes which can contribute to the kernel row number. So that we took all the available genes till today, and we try to characterize our genotypes for whether these particular genes are available or whether they are expressing in our genotype or not. So in that way, we could found that whatever the genes we have taken, these are from temperate background. We are studying in our tropical background. So whether these genes are, of course, they showed associated with high kernel row number. Means this they show the signal when we have the high kernel row genotype. Low kernel row genotypes also, that there are some signals, but majority of them are present only in high kernel row number genotypes. These were we further that particular genes where we further converted into allele specific markers. Allele specific marker, how we sequence this particular genotype, high and low kind of genotypes, where there is a polymorphic site observed between high and low kernel genotype, that is 36 and 37 we can say. So for example, in this case, 35 is the low kernel row number genotype, and 36 is the high kernel row number genotype. We can see the polymorphic region available in the sequences. So using this, we decide and we develop the polymorphic, allele specific markers, and these markers are further utilized for identification of these genotypes and also for the hybrid breeding. So this is one our understanding with respect to kn genes, kernel row number genes. Our main topic is like cob length. I'm coming to that, because this kn as well as cob length are little contradictory. Wherever kernel row number is increased, cob girth is increased, but length going to be reduced. So our target is our genotype should have more kernel row number as well as cob length. Then only we can pack good amount of seeds and go for the better productivity. So before that, that's why we first we understood the kernel row number genes, how they are operating in our genotype, and perfectly we developed some of the markers which can see you can differentiate 35 from the 36. The 35 there is no marker signal, and 36 there is amplification of this particular marker. The same way 40 this 5 and 42. 42 you can see maybe while we let the visibilities were less, but the 42 is having that particular marker, and 5 is having no uh, don't have that marker. This 5 as well as 35 are low in kernel row number, and 36 and 42 are the high kernel row number. So with this, we understood that these genes are also present working in our genotypes. Present, of course, they will be presented, but these markers can clearly differentiated our tropical background genotypes for the kernel row number is concerned. So later on, then as I told you, the cob length is our main target. So if the cob length is increased, then we can, with the increased kernel row numbers, with the cob length also increased, so more number of kernels can be fit into that, so that indirectly yield can be increased. That was our motto. So then we try to understand the cob length also. Cob length, there are different genes are responsible for cob length. So kn1 gene is one of the important genes, major genes responsible for gibberellic acid pathway. Gibberellic acid, like the effect of gibberellic acid, where it is synthesized on those things. Gibberellic acid pathway is finally decided. The kn1 is deciding the controlling the gibberellic acid composition in cobs also. So we took that particular kn1 gene for understanding the cob length. So this kn1 that encodes gibberellic acid to oxidase or GA2 oxidase, and that will be having controlling both pathway, and then finally it regulates both all cob traits as well as kn. So here this kn1 also fortunately controls kn also, as well as cob length traits also. We try to understood this gene for developing our hybrids further. So here also we did same thing. Preliminary evaluation we did whatever the germplasm available at IRI, that were ranges of cob length were there. Earlier I showed it is the ranges of kernel row numbers were there. Now the same phenotypes, same population we studied, there was a ranges of cob length from 7-8 centimeter to up to 20 centimeters. So we categorize this into low cob length category and as well as high cob length category. So see, you can see there is plenty of variability available up to starting from 5, 6 to 8 centimeter, 21, 21, 30 also is length of the cob. So then we understand that with genetics part of this, we could identify that these states are less affected by environment, and there more of genetic control is there, and these states are heritable. These both trait for that matter, kernel row number as well as cob length. The heritability is very much important, whether it is expressing and going contributing the next generation, or if it is going what type of gene action is moving forward. These kind of studies we did, and we found that these two traits are very highly heritable. So taking this, again we did the same thing what we did in the kernel row number. We took different kinds of genes. The knotted one genes, which is mostly present in the present on the chromosome number 1, that will be that will have dissected, and we came to know that here also there is a good correlation with where there is a kn1 gene is there, that the cob length approximately the length of the cob was more. And similarly TD1 and IDS1, I'm not going to discuss this, but this is the one gene which we are targeting, that kn1. There is a presence of this gene in our tropical background also, of course, and this were going or having some tendency to go with high cob length genotypes. So that means wherever high cob length is there, these genes tend to present, or these genes show their presence. Okay. Further, what we did the next generation sequence based approach to find out the allelic variation again, or the nucleotide level difference at nucleotides level or amino acid level. So we sequence this in using next generation sequencing method, and these data were cured on alignments were made. I will show in the next slide. Alignments were made between high and low cob length genotype, and we could find there is a perfect, again as we expected in the kn genotype, there is a polymorphic site between the high and low cob length genotypes. B73 is our reference genotype. I think you might have Now, what is the importance of the reference genotype? These reference genotype already genes were coded or decoded, sorry, and all the annotations were done, and we aligned our uh, uh, these sequences with reference genotypes and also among the high and low covalent genotypes. We good amount of variations, and we try to catch these variations further. So these are the total 64 SNPs were observed. This we call it as SNP, which is generated because of the polymorphic site between the high and low covalent genotype. That is a variation with respect to the particular nucleotide site. These are the SNPs what we have observed at particular positions. So for the students, for the sake of student, I can say we have to count each individual nucleotides and their position. What is happening with respect to genotype 1, genotype 2, genotype 3, genotype 4, and we could get some of the genotype. There is a variation at uh, amino acid level also. Sometimes there is a no amino acid changes. If there is amino acid changes, this we are call it as a non-synonymous mutation. If there is no, it is a synonymous mutation. Though there is a changes at nucleotide level. In that way, we could find around 13 SNPs which were causing non-synonymous mutation. Non-synonymous mutation in the sense they are causing changes at the amino acid level also. So what we did was like we tried to screen this genotype with the marker we have designed for this also, but we failed to differentiate this genotype with the marker. We failed to differentiate with the particular marker. Earlier cases, we were successfully differentiating high kn genotypes and low kn genotypes, but in this case, covalent with respect to covalent, we could not differentiate it. But our thinking was like there will be some changes. Otherwise, this SNP will certainly causing some changes, but with marker we may not capture, but there will be some certain changes. To know that, we went for the CDS alignment, from CDS alignment. So in that we could identify all the CDSs were like changes were not present between high covalent. This is the high covalent genotype. This was the low covalent genotype. See, like this in pictorial depiction also you can see this was the high covalent genotype and this was the low covalent, sorry, covalent genotype. So for at 8th CDS there is a deletion. There is a deletion of some particular amino acids. So around 75 amino acids were deleted at a particular 8th CDS. Okay. So that we concluded that though the marker could not identify or differentiate it, I think there may be little more effort is required. But we clearly understood that this changes at amino acid level at 8th CDS. That the total 75 nucleotide difference was there. That will lead to around 25 amino acid changes. The mRNA length was different. So this might have caused this variation in the kernel covalent in our genotype. So to test this, we made some homology modeling between low and high covalent genes. This is 541 is the high covalent and 542 is the low COP comparatively. So then there is a difference in the gene or protein coding for this particular genotypes also. At NOX3, in case of AI542, it homeobox protein not at like 6, it is responsible for deciding covalent in 541. 541 is covalent is more. That may be due to like negative effect of that. Like some part is deleted. The deletion might have be favorable for increasing the covalent in case of 541. 541. So later on, we did Ramachandra plot also to identify where exactly that particular changes are happening. We can see that there is a changes at particular site, that is site number 1, site number 2. There are so many changes with respect to this particular NOX family genotype with respect to high covalent genotype as well as low covalent genotype. Now we understood that there is a changes, there is a deletion, some nucleotide, 75 nucleotide or 25 amino acid changes that might have caused this variation in the covalent among the tested genotypes. Further expression analysis were done. Expression analysis were done. This was the covalent, high covalent genotypes compared to this. This was the low covalent genotype. Expression analysis were done with their hybrids also. We crossed these two and we made the hybrid. the genes which have that deleted part. For the deleted part, we designed a primer and expression analysis were done, and then you could see that the AI541, the cov length was high, the expression of the gene also very high, and the AI542, the cov length was low, the expression of gene was also less, and where hybrid is intermediary, like it is having additive kind of action. So this we understood that the both goes together. They will increase not exactly to the parent. It is just like in additive mode. Then hybrid also have the advantage of increasing cob length. So cob length as well as along with the cob girth, as earlier I told you, that the cob girth simultaneously, if you can put and breed for the hybrids, the hybrid will be having high productivity. So like that, we have developed so many good hybrids with having more number of kernels. You can see this is so big, girth is so high, and also as well as the length of the cob. So in this way, we can accumulate traits which are component traits which are indirectly deciding the productivity to increase the productivity of the corn. So these were the hybrid what I have shown. All these hybrids which having high productivity of more than 10 tons per hectare were demonstrated to the higher authorities, and even Minister of Agriculture also visited this AI 4271, that what the one which is I have shown here, so which is having more than 10 tons, around 10 tons per hectare yield, or visited by the farmers also, ministers also, and should be well appreciated. So finally, I can conclude this like the kn1 gene, which is one of the important gene which decides Hoblen, they may have some of the mutations accumulated in case of tropical maize, that were some 25 nucleotide differences there, amino acid differences there, that can be further dissected and marker can be designed for that. And resulted of amino acid changes might have caused a novel function of KN1 that has to be further also be studied. And KN1 induces cell purification, so that you have already understood that it is involving in gibberellic acid pathway that might have increased Coblen. And Coblen showed additive gene action that the hybrid studies we have decided. And this 25 amino acids, those changes, what exactly happens at expression level or what we need to study further. So this is the utilization of some of the component traits, or how can we dissect and improve the yield in maize. That is the major target task we have taking care, and further we are going in mm, similar way. We are going for the other traits also, for the genetic enhancement of grain in case of maize. So with this, I thank organizers for giving this opportunity, and also Director IRI, Head of the Genetics, and our staffs, and ICR for um, the facility, and then the project which we work in SCRB, DST. So all are acknowledging. Thank you for your patience hearing, and if any